Hello to all the VGRI workshop attendees who are watching my talk right now. I'm Carolina Rivera, a member of the Global Wheat Program at CIMMYT. I am very happy to present my research to you today, and as Anna said, I will talk to you about sourcing balance and estimations of organ contribution to grain weight in wheat. But what is actually sourcing balance? Before we start, we need to know some very important concepts. Source can be organs such as leaves or any other photosynthetic tissue, or can be also traits such as photosynthesis or light interception. And we define them as source conceptually since they provide a simulate for the zinc organs. Zinc traits or organs receive these photoassimilate from the source for growing purposes and are more straightforward to identify since they are related mainly to grains, for example, grain weight, grain number, or any other yield component. Now, yield can be limited by source or by zinc during grain filling, or can be limited by both, which is known as a co-limitation. Sourcing balance is simply the difference between the potential amount of assimilate available for grain filling and the capacity of the grains to store it. And finally, the most common methodology to estimate the status of yield limitation is through sourcing manipulation treatments. For example, degraining, that is removing half of the grains in the spikes before the onset of grain filling. To give you some literature background, wheat yield under favorable conditions is normally defined as zinc limited during grain filling. Experiments where extra light treatments were imposed during pre-anthesis resulted in a higher grain number, and this higher grain number at the same time increased the photosynthetic rate during grain filling periods, indicating that while source limits grain pre-anthesis, zincs limit the realization of photosynthetic capacity post-anthesis. However, there is evidence that in modern cultivars, grain growth may be limited by both source and zinc during grain filling. And this would mean that simultaneous increases of source and zinc size will be a crucial task in breeding for higher yield potential. Within this context, our first research question is, what is the source-zinc balance status of elite CIMMYT spring wheat cultivars? Is there a difference between older versus modern cultivars? And when we are talking about improving source strength and photosynthesis contribution during grain filling, we need to talk about organs that are involved in supplying assimilate for grain growth, organs such as leaves, spikes, and stems. So our second question was related to photosynthetic contributions of leaves and stems to final grain weight. And finally, is it possible to upregulate photosynthesis and pre-anthesis reserve translocation to grain in response to artificial reductions in source? To answer these questions, we conducted field trials during 4 consecutive years from 2011 to 2014 under yield potential conditions, where we evaluated 26 elite CIMMYT spring wheat cultivars, mainly high yield releases which have been grown worldwide. We applied 3 treatments 10 days after anthesis in 12 shoots per treatment. The first was degraining, the far left image. We did this in 26 cultivars. Defoliation of the top 3 leaves, and the picture in the center. We did this in 9 cultivars, and shading stems with a piece of textile that allowed for normal transpiration. You can see this in the far right image. We applied this in just 4 cultivars. And we also tagged 12 unmanipulated shoots as controls. The traits and responses to treatments we evaluated were final grain weight, the contribution and translocation efficiency of stem pre-anthesis reserves, and punctual measurements of spike and flag leaf photosynthesis during grain filling. We also measure light interception and green area index at anthesis in control shoots to estimate reductions of these 2 traits by defoliation. The way we interpret results from degraining treatments is that we have the same availability of assimilates for 50% fewer grains. So if final grain weight remains unchanged or increase up to 10%, we can talk about a zinc limitation. If grain weight is increased from 10% to 30%, we are talking about source or co-limitation. For defoliation or shading treatments, it's more simple. We just estimate the contribution of the organ by the percentage decrease in final grain weight in defoliated or shaded shoots. Results from degraining treatments showed a wide range of grain weight responses. In the left-hand side graph, we can observe that as further the points are from the one-to-one line, the bigger is the increase in grain weight, reflecting more source limitation. If the cultivars are closer to the one-to-one line, they are more zinc limited during refilling. And across the 26 cultivars, we observed an average of 11% increase in grain weight, ranging from no response or complete zinc limitation to increases in grain weight of nearly 30%, indicating a more source limited scenario. In the right-hand side figure, we plotted the grain weight response versus the year of release of the cultivar, where we can see a trend for more modern cultivars to show a higher grain weight response, indicating that there is a higher extent of source limitation in more modern cultivars where releases cultivars, but since most of the cultivars were released from 2000 to 2009, we suggest to confirm this trend with landmark cultivars with a wider range of year of release. Comparing control and defoliated shoots, we estimated reductions in light interception due to the removal of the top 3 leaves at 62% and reductions in grain area index by 49%. The contribution of the stem photosynthesis to grain weight during grain filling were estimated at 10% in average, with no difference across the subset of 4 cultivars, probably linked to the very similar stem area for these cultivars. But these numbers can be comparable to previous estimates for flag leaf contribution from 5 to 10% under irrigated conditions. So it has been suggested that stem plus leaf sheath photosynthesis may be especially relevant during the second half of grain filling when temperature is typically higher and leaf blades have started to senesce. Considering reductions in light interception by 60% and green area index by nearly 50% due to removal of the top 3 leaves, reductions in grain weight due to defoliation were conservative, averaging 17% across a subset of 9 cultivars, and showing significant differences among cultivars ranging from 10 to 20%. The relatively small reduction in grain weight in defoliated shoots could be partly linked to the increased contribution of stem pre-anthesis reserves or linked to an upregulation of photosynthesis in other organs, so we investigated these mechanisms. But it was not possible to detect spike or flag leaf photosynthesis responses from survey or punctual photosynthesis measurements in treated shoots. This was probably related to the nature of the measurements, since survey measurements might not reflect the overall photosynthetic rates during grain filling. And although these results suggest no signaling mechanisms induce increases in leaf or spike photosynthesis, we do not discard the possibility since certain extent of co-limitation by zinc and source was confirmed by the grain weight reductions we saw from the treatments. Lastly, although we did not see significant responses in spike or flag leaf photosynthesis, the contribution of stem pre-anthesis reserves to final grain weight was doubled in defoliated shoots in comparison to control shoots. So the plant somehow had to compensate for losses in source strength or photosynthesis during grain filling, and allocating more stem pre-anthesis reserves appeared to be one of the mechanisms to achieve that. In conclusion, degraining increased grain weight between 0 to 28%, suggesting both zinc and co-limitation in CIMMYT elite spring wheat lines, where modern lines appear to be more limited by source than older lines. And even when extreme reductions in light interception of 60% during grain filling, grain weight reduction was moderate, in average of 17%. Stem photosynthesis contributed 10% in average to final grain weight during grain filling, similar to previous estimates of flag leaf contributions, indicating stem photosynthesis plays a key role during grain filling, and this might be especially important when the second half of grain filling is source limited or under drought stress when leaves can senesce prematurely. We did not detect photosynthesis upregulation in response to reductions in source with survey measurements, but the contribution of pre-anthesis reserves was doubled. Overall, it is suggested that increasing photosynthetic capacity post-anthesis through increases in pre-anthesis rhizomatic contribution and translocation efficiency, and also enhancing stem and leaf sheath photosynthesis may play a role in avoiding source limitation in future wheat breeding for yield potential. And that is all for me. Thank you for your attention, and I am now open to questions. Thank you.